Okay, my name is Joan Luby, and I'm giving the talk in the symposium on economics and emotion. And my talk is about early experience in brain development, informing a new science of neurodevelopmental optimization. And the key principles of this talk are that we know that experience materially impacts brain development. We know this because there's strong associations between experience and brain development, and that have been demonstrated in multiple longitudinal human studies. We know that there's causality between experience and brain development that's been demonstrated in animal models. And we know that this causality has been shown in several enhancement studies in institutionalized children. The impact of experience on the brain is particularly powerful early in development during sensitive periods. So the entire thrust of this talk has to do with how these sensitive periods should inform public health planning to optimize child neurodevelopment in early childhood, and the idea that empirically informing when sensitive periods arise for various skills and abilities, and cognitive and emotional and otherwise, can be used to know when to protect children optimally at certain times or when to enhance their development at certain times. So all of these principles are designed to basically create an empirically informed early developmental health promotion or health enhancement program. And to date, there's surprisingly little attention to methods to enhance development early in life. And an understanding of risk trajectories and sensitive periods can inform when it's most important for children to be protected from risk or exposed to stimuli or challenges to optimize development. This is currently driven by just sort of hearsay and old wives' tales, but it actually can be much more informed by empirical data and needs to be. So a model system for how experience influences development is in the immune system. The immune system is the singular organ of the body that specializes in responding to environmental exposures. And the immune system knows how to differentiate things that are inert from things that are toxic versus innocuous and dangerous. And the capacity of the system to perform this task develops in response to early exposures in early childhood. And this is, of course, why vaccines are given in early childhood. So in that sense, the immune system provides an ideal model for how a biological system is informed by experience to the advantage and the detriment of later health. And the immune system is one of the key consummate developmental origins of health and disease systems. This is really a principle that asserts that there's important individual differences in disease susceptibility that are correlated with differences in immune development. The developmental origins of health and disease posits that adult disease can be traced back to childhood, perinatal exposures, or even in utero exposures. And the responses to environmental exposures confer short-term survival, but also have lasting implications for immune development and later health. So the immune system is the consummate developmental origins of health and disease system. And it includes, and it's a parallel that early life programming directs later immune health through epigenetic mechanisms. And we know this is also true in brain development. So one example of developmental origins of health and disease with the immune system are the babies that are born in fall or winter and therefore are kept inside in the northern hemisphere and are kept inside, don't have a lot of sun exposure, then have inadequate vitamin D synthesis, they have higher rates of eczema, they have excessive antigen exposure, and this results in greater sensitization and higher food allergies. So we know that allergies are more common in babies born in the northern hemisphere in fall and winter related to differential exposures and how the immune system then responds to that. So we know a decent amount about how the brain responds to early experience, and that literature has been burgeoning over the past 15 to 10 to 15 years. But there are some specific brain regions that are particularly well established to have relationships to early experience. The hippocampus is one, and we know from a number of studies that the hippocampus has decreased volume related to poverty, related to low maternal support, related to experience of abuse, and related to adverse childhood experiences more generally. We also know that there's regional volume loss in rats who are exposed to adversity, and we know here in these animal models that this is based on stunting of dendritic growth and pruning. We also know there's decreased connectivity associated with early adversity, but not with deprivation from institutional care. The amygdala is also a region that we know to be impacted by early experience. There is reduced amygdala volume associated with adversity, increased reactivity to threat associated with adversity, and reduced connectivity to the control regions of the VMPFC. The striatum, the area in which reward processing takes place, has also been established to be responsive to experience. We know that neglect and maltreatment is associated with reduced activation of striatal regions in response to reward anticipation and receipt. We know that early poverty is associated with reduced volume of the caudate, a region of the striatum, and we know that rodent models show that poverty produces a decreased ability to experience reward or anhedonic-like states with aberrant structure of reward circuitry in rats observed using fMRI. We also know that prefrontal cortical regions are impacted by experience and particularly stress and adversity. So children exposed to deprivation or have decreased volume and thickness of the dorsolateral prefrontal cortex. The other question has to do with how adversity has specific effects on brain regions, both not only regional specificity, but also timing specificity. And this is one of the key issues that needs to be mapped out in order to develop this neurodevelopmental optimization program. So we've shown in some of our studies that there is regional specificity for both the effects of adversity and support. The hippocampus is one region that's impacted by adversity in the preschool period and maternal support during the school age period. Of course, negatively impacted by adversity and positively impacted by maternal support. The amygdala shows a very different pattern of response to experience, where preschool ACEs and high maternal support have different impacts on the trajectory of the amygdala. And then the caudate, an entirely unique trajectory where there's essentially an additive effect of ACEs and maternal support during the preschool period and the school age period. So in addition to regional specificity, we also have to look at timing specificity. So developmental data is now needed to inform how the effects of the psychosocial environment, broadly defined, impact brain and behavioral development. This is necessary to create empirically informed guidelines for when it's most important to protect children from adversity and when to expose them to challenges. There's regional specificity in these exposures, as I've just shown, but there may also be timing specificity as well. In addition to regional and timing specificity, another question has to do with the nature of the experience. There's been decent literature showing that experiences of deprivation versus experiences of threat have a different impact on brain development. Experience of disruption in the caregiving relationship, such as that found in institutionalized children, has a different effect. And the question of whether different types of enhancement impact different brain regions differently at different times. There are animal models that have explored causality in the relationship between adversity and brain development. And these animal models are necessary to explore causality because in humans, obviously, we can only look at associations outside of those few studies that have looked at institutionalized children and have used enhancement paradigms. But what I'm arguing for is actually the use of enhancement paradigms in human samples, which could begin to establish causality. But several experimental animal paradigms simulate early life adversity. These are separation from the dam either once or chronically, and this models the impacts of disruptions of maternal care on the infant brain. Poverty can be simulated in animal models by creating a resource scarce environment. Resource scarcity manipulations, however, also influence maternal care patterns. So you can't just have resource scarcity with optimal caregiving, but this is also what happens in human samples. When there's resource scarcity, it impacts caregiving in a detrimental way. And those animals who are exposed to resource scarcity generally have mothers who have chaotic or abusive parenting approaches. Maternal deprivation and abnormal maternal care have been studied in non-human primates, enabling examination of salient brain regions. In rat models, those who are exposed to low bedding or resource scarcity compared to controls basically have mothers who show more chaotic nurturing. So they have high levels of what has been termed entropy. Those mothers have unpredictable parenting. They tend to frenetically run around the cage searching for resources, as compared to controls that have ample resources that do a lot more nurturance and snuggling behavior with their infants. Similarly, we also know that rats who are exposed to adversity, based on these low resource, low bedding paradigms, have decreased play as well, suggesting that the babies who are exposed to adversity show an anhedonic-like behavior. These are all studies coming out of a lab at UC Davis, Tally Barham. And she's also shown that these unpredictable maternal behaviors are found not only in animals, but also in humans, and that when these unpredictable maternal behaviors occur during sensitive periods in development, they tend to have long-lasting impacts on brain and cognitive development later in life, even if nurturing maternal care is restored, suggesting there's a sensitive period. So the proposed developmental enhancement or optimization paradigm is really described here in this slide. And it's the idea that children reared in adverse circumstances versus optimal circumstances and have different brain developmental outcomes based on whether the adversity or the enhancement occurs during a sensitive period. And it also suggests that in a developmental optimization paradigm, children might need to be protected from adversity, particularly during certain periods of time. They might need less protection at other periods of time, and then they might be most responsive to enhancement during certain periods of development as well, based on sensitive periods. So this is a theoretical model that suggests that empirical data in animal models and in human studies that use enhancement studies might be able to map out sensitive periods based on RDoC type skill sets, and might look at when it's most key to protect children from adversity. While of course we would aspire to always rear children in the most optimal circumstances and have the least exposure to adversity, we know that that's not possible for more than half of the world's children, which is why a more nuanced and empirically informed optimization program is necessary. So the development of the immune system is really a model for this neurodevelopmental enhancement system. And it's the idea of the brain interacting with the environment in a way that optimizes brain development. So as mentioned, the immune system is one system, a biological system that distinguishes friend from foe when interacting with the environment. It develops a memory of foe or toxins by developing antigens so that it can protect the organism from these things in the future. The immune system also plays a role in neurodevelopment in the sense that microglia is the immune cell of the brain, which is a macrophage. It's known to be highly responsive to environmental influences, as are all immune cells. It has three phases of growth, and it's influenced by the prenatal environment. There's sex differences in response to environmental factors that many speculate might be a source of sexual dimorphism in many neuropsychiatric disorders, such as autism or ADHD. There's also links between the microbiome and microglia play an important role in pruning, which of course is also one of the processes known to be very, very responsive to environmental influences. And one parallel line of literature has looked at the response of the immune system to poverty and adversity or low levels of maternal warmth. And we know that children who experience low levels of maternal warmth and children reared in poverty tend to have a pro-inflammatory phenotype where the immune system is activated in a chronic and non-healthy way in response to these environmental adversities. So one question is, how might we optimize immune development? When is it best to expose children to doses of pathogens to enhance immunity? This is a similar question to when are immunizations best well-timed? And this is actually surprisingly not as well informed by empirical data from human samples as you might think. Little is known about the development of the immune system, how it interacts with environmental exposures in a very specific way, and there are clear psychosocial correlates of immune response, as I just mentioned, the pro-inflammatory phenotype. Other factors that are low-hanging fruit to look at the effects of experience on brain development are, for example, things like sleep and how sleep influences neurodevelopment. We know that sleep in childhood definitely influences brain development. The proportions of slow wave activity is very much related to synaptic density, and this changes across early development, but there's still much that is not known about the impact of sleep on neurodevelopment. And the more we knew about that, the more we could make very specific recommendations about sleep hygiene in early childhood. So this is just an example of some of the things that are known, that timely sleep facilitates declarative memory consolidation in infants, and sleep and learning and plasticity are very much impacted, but exactly how this is related to brain development using fMRI and at the cellular level, of course, remains unknown. So the objective here is the idea of optimizing brain development, pulling on the idea of the data that we know about experience in brain development, and then making this a much more fine-grained and nuanced database by also factoring in sensitive periods. So it's the question of when exposures to nurturing caregivers is most critical, when avoidance of separation from caregivers is most critical. We really still don't understand that. How do we educate caregivers and develop a more nuanced guide to caregiving? What type of caregiving is most important? What type of caregiving is most important in temperamentally anxious children, for example? When are the sensitive periods for this? Are there key periods when protection from adversity is critical? And are there key psychosocial exposures, like exposure to language? We know this is a sensitive period, like exposure to social contact, different types of sleep hygiene, exposure to peers that might be most optimal to enhance brain development. So the next steps for this new science of neurodevelopmental optimization are enhancement studies in humans. Because of course we don't want to and can't expose humans to deprivations, we can expose them to enhancement. So these studies could enact randomized controlled trials that provide skill enhancement in specific domains at specific times in life, or parenting enhancement at different periods in development. We can look at its impact on neural outcomes, and we can also look at effect sizes to see whether there are in fact sensitive periods. Parallel models can be done in animal studies to look at processes on the cellular and molecular level. And this is just a slide showing an ongoing study that we have where we're collecting a sample of infants where we followed the mothers in pregnancy, and we've looked at both immune factors in pregnancy, psychosocial stress in pregnancy, depression in pregnancy, and then we're looking at neurodevelopment in the infants, inflammatory markers in the infants from birth to age 3, as well as the gut microbiome, which we believe has some relationship to neurodevelopment, not yet well understood, and certainly does have a relationship to immune development. But the question is of how all of these environmental factors influence neurodevelopment to give us a foundation to begin to develop this model of early neurodevelopmental enhancement. Thanks for listening.
Is it over?
It's over.